A groundbreaking discovery has emerged in the world of entomology. A new species of wasp has been identified in the United States for the very first time. While oak gall wasps and their predators may not possess the vibrant allure of butterflies, they are increasingly capturing the attention of both scientists and nature enthusiasts alike. These tiny insects, measuring between 1 to 8 millimeters in length, are responsible for creating galls, tumor-like growths on plants. These galls can vary dramatically in size, from as small as a pinhead to as large as an apple, and they can take on fascinating shapes, some resembling sea urchins or saucers. This intriguing phenomenon was explained by Kirsten Pryor, an associate professor of biological sciences at Binghamton University, who also co-directs the university's Natural Global Environmental Change Center. If there's one thing these wasps symbolize, it's the rich tapestry of biodiversity. North America is home to approximately 90 species of oak trees, which in turn host around 800 species of oak gall wasps. Interestingly, parasitic wasps lay their eggs within these galls, ultimately consuming the oak gall wasps themselves. But just how many species of these parasitic wasps exist? This is a question that both academic researchers and everyday citizens are eager to explore. A recent publication in the Journal of Hymenoptera Research, titled Discovery of Two Palearctic Butanomia Geralt Hymenoptera megastigmidae Parasitic Wasp Species Introduced to North America, sheds light on a previously unrecognized level of species diversity. The article features contributions from Pryor, current graduate student Kathy Friedrich, former graduate student Dylan G. Jones, and several researchers from the University of Iowa, as well as Elijah Goodwin from the Stone Barn Center for Food and Agriculture in Tarrytown, New York. This discovery is part of a broader research initiative. In 2024, Binghamton University received a substantial grant of $305,209 from the National Science Foundation, NSF, to investigate the diversity of oak gall wasps and their parasitoids across North America. This project is a collaborative effort involving Pryor, Andrew Fords from the University of Iowa, Glenn Hood from Wayne State University, and Adam Kranz, one of the creators of gallformers.org, a website dedicated to educating the public about galls found on North American plants. The NSF grant aims to address a fundamental question: how do gall-forming insects evade the diverse and evolving groups of parasitic wasps? And conversely, how do these parasites adapt? To tackle this question, researchers are gathering oak gall wasps from various locations across North America and employing genetic sequencing techniques to identify which parasitic wasps emerge from the galls. Among the researchers are Friedrich and fellow Binghamton graduate student Zachary Preet, who spent their summer collecting galls and parasitoids from New York to Florida. We are keen to understand how the characteristics of oak galls serve as defenses against parasites and how they influence the evolutionary paths of both oak gall wasps and their parasitic counterparts. The scale of this study will make it the most comprehensive co-phylogenetic study of its kind. Pryor stated, "Only through a concerted effort to explore biodiversity can we uncover unexpected findings, such as new or introduced species, unveiling unknown species." In recent years, researchers from Pryor's lab have traveled along the West Coast from California to British Columbia, collecting around 25 species of oak gall wasps and rearing tens of thousands of parasitic wasps, which were ultimately classified into over 100 distinct species. Some of the parasitoids reared from various oak gall wasp species turned out to be the European species Butanomia dorsalis, belonging to the Megastigmidae family. Researchers at the University of Iowa also identified a similar wasp from their collections in New York. The discovery of this potential European species on both coasts of North America motivated our team to verify the identity of this parasitic species and determine whether it was indeed an introduced species from Europe, Pryor elaborated. Identifying parasitic wasps can be quite challenging due to their small size and subtle features. To overcome this, researchers utilize genetic tools to confirm species identities by sequencing a gene known as the universal barcoding gene, cytochrome oxidase subunit I, MTCOI, and comparing their findings to established reference libraries. Their research revealed that the European species B. dorsalis exists in two distinct varieties, or clades. The samples from New York were genetically linked to species found in Portugal, Iran, and Italy, while those from the Pacific coast were related to wasps from Spain, Hungary, and Iran. The genetic sequences from these two clades were sufficiently different to classify them as separate species. This indicates that B. dorsalis was introduced to North America at least twice, suggesting that the introductions on the east and west coasts occurred independently, Pryor noted. Interestingly, while B. dorsalis was found in at least 4 different oak gall wasp species from Oregon to British Columbia, all the West Coast specimens were genetically identical, indicating a localized and limited introduction. In contrast, the East Coast wasps exhibited slightly greater genetic diversity, which may suggest a less pronounced population bottleneck or multiple introductions of the species. How did these European wasps make their way to North America? One theory is that non-native oak species were deliberately brought over. The English oak, or Quercus robur, has been cultivated for timber since the 17th century and can be found in British Columbia as well as several northeastern states. The Turkey oak, Quercus cerris, is another ornamental tree that has established itself along the East Coast, including near the location where B. dorsalis was discovered in New York. There are other potential explanations as well. Adult parasitic wasps can live for up to 27 days, which means they could have inadvertently traveled on an airplane, according to Pryor. At this point, researchers are still uncertain whether these introduced species pose a threat to native North American species. It is known that other introduced parasitic species have negatively impacted populations of native insects. She acknowledged, "We have observed that they can parasitize multiple oak gall wasp species, and that they have the potential to spread. Given that the population on the West Coast likely expanded across regions and host species from a small initial introduction," Pryor explained, "they could be influencing the populations of native oak gall wasp species or other native parasites that target oak gall wasps." The role of naturalists and citizen scientists is crucial in biodiversity research, as demonstrated by the project that led to the identification of the 2 B. dorsalis clades. Gull Week, an initiative hosted on the iNaturalist platform, encourages citizen scientists to collect galls during 2 specific seasons, and specimens from the NSF-funded study will be shared on the naturalist site gullformers.org. Binghamton University's ecology classes have actively participated in Gull Week and have also documented gulls during the university's annual EcoBlitz biodiversity event. Biodiversity is essential for maintaining healthy and functioning ecosystems, yet it faces increasing threats from global changes. Parasitic wasps are likely the most diverse group of animals on Earth and play a vital role in ecological systems, acting as biological control agents that help regulate insect populations, including those that are pests in crops or forests, Pryor emphasized.